Welcome back, everyone. This is part C of our series on the electron transport chain. This mini lecture will focus on understanding the complexes involved in the electron transport chain more. So to begin, let's talk about complex 1, which is the NADH dehydrogenase. This is the beginning of electron transport, where we transport the electrons, the hydrogen protons, and the electrons from NADH to complex 1. The loss of the hydrogen when we oxidize NADH helps regenerate the NAD that is needed for the citric acid cycle. Remember, this reaction is happening on the matrix side, which means that the NAD that is generated from the oxidation of NADH can be used immediately by the citric acid cycle, as NAD is a substrate needed, it's that coenzyme needed for many of the reactions of the citric acid cycle. Hydrogen ions and the electrons are transferred to the mobile electron acceptor, which is ubiquitone or coenzyme Q, and this forms coenzyme Q with 2 hydrogen ions. The coenzyme Q, H2, carries these electrons from complex 1 and 2 to complex 3. As you can see from this picture on complex 1, it is extremely complex. It is a quaternary protein. That means it has multiple subunits involved. It also is an enzyme, so it catalyzes reactions. And the reaction is catalyzing is the oxidation of NADH. So you can see it here, NADH plus a hydrogen proton plus the coenzyme Q leads to NAD plus the coenzyme Q with 2 hydrogen protons. And so for every NADH that's oxidized, 2 electrons are transferred to coenzyme Q, and 4 hydrogen protons are pumped through complex 1. Because the passing of these electrons from NADH to the coenzyme Q releases free energy that allows 4 hydrogen protons to be pumped across the membrane, across its concentration gradient to the intermembrane space. Complex 2 is a smaller complex that does not span the membrane. And so ubiquitone, or coenzyme Q, obtains the hydrogen ions and the electrons directly from FADH2 and becomes coenzyme QH2 to bring these electrons to complex 3. The 2 electrons are transferred to the mobile carrier ubiquitone through the reaction below. So we have FADH2 combining with ubiquitone, or coenzyme Q, leading to the products which are FAD plus coenzyme QH2. So we've transferred the electrons to the ubiquitone, coenzyme Q with H2 now. Now the FAD can go back to the citric acid cycle and act as that coenzyme substrate that's needed for the running of the citric acid cycle. The next complex is complex 3, cytochrome BC1. And this complex does span the entire membrane, so it is a large membrane protein. Remember, it also is an enzyme. So it receives 2 electrons from ubiquitone, or coenzyme Q, and it also contains ferritin, also known as iron. So it contains a cofactor here, which is reduced to Fe2+ and oxidized to Fe3+, which means 2 hydrogen atoms. So what it does is it generates energy from electron transfer to pump 4 hydrogen protons from the matrix to the intermembrane space, increasing the hydrogen ion gradient. And here's the reaction here. We have ubiquitone with 2 hydrogen protons plus 2 cytochrome C. The iron is in its oxidized state. Then the products are coenzyme Q, or ubiquitone, plus 2 hydrogen protons, or electrons in this case, plus 2 cytochrome C and the ferritin in a reduced state. Now the last complex is complex 4. This is a large complex as well. Remember, it's an enzyme complex, it's a protein complex, and it spans the entire membrane. We have 4 electrons from cytochrome C are being passed to oxygen, which is the last electron acceptor. The electrons combine with hydrogen ions and oxygen to form 2 molecules of water. So the reaction is 2 electrons plus 2 hydrogen protons plus a half of an oxygen molecule leads to 1 molecule of water. Typically this happens in double, so we would see 4 electrons combining with 4 hydrogen protons combining with 1 molecule of oxygen, leading to 2 molecules of water, just to make it a little bit more simple and even. So the energy is used to pump 2 hydrogen protons from the mitochondrial matrix into the inner membrane space, further increasing the hydrogen ion gradient. Remember, we pump 2 hydrogen protons at this step across the membrane. It is very important to note that this is the only step in the whole process of glucose oxidation where we see oxygen, that it acts as the last electron acceptor. This is very important because if we do not have enough oxygen, complex 4 would not run and the whole electron transport system would not operate. And so this step, having enough oxygen, is vital in order for each step of the complex to run. Here is the diagram of complex 4. As you can see, it is a very large quaternary protein as an enzyme. That means that we have multiple subunits. You see subunit 1, subunit 2, subunit 3. We also see some cofactors involved. We see ferritin, or Fe, commonly known as iron. We also see copper, denoted as Cu. So there are important cofactors in order for this enzyme to run. Hence the reason why microminerals are important in our diet, and they are essential because they're needed in these complexes of the electron transport chain. What we see here is we see 2 hydrogen protons combining with an oxygen molecule. In this case, it would be half of an oxygen to yield water. We also see 2 hydrogen ions or protons being pumped across the membrane, enhancing the proton gradient. Okay, so remember, the whole process of electron transport is to release free energy for which we can then pump hydrogen protons against its concentration gradient into the intermembrane space and produce something called a hydrogen or proton gradient. Now I would like to return to this fairly simple illustration of the electron transport chain. You can see the 4 complexes, complex 1, 2, 3, and 4. You can see complex 1, 3, and 4 that span the membrane. We can see ubiquitone, or Q, and cytochrome C as our mobile electron carriers. What I want to draw your attention to is the number of hydrogen protons that are pumped per complex. So complex 1 pumps 4 hydrogen protons from the oxidation of NADH. Complex 3 pumps 4 hydrogen protons because of the electrons that are carried from ubiquitone. And complex 4 transports 2 hydrogen protons across the membrane. So in total, we have 10 hydrogen protons pumped across the membrane. Now, how many are contributed from NADH and FADH2 independently? So if we look at NADH, we have the 4 being pumped across from its oxidation in complex 1. It contributes to the 4 in complex 3, and it contributes to the pumping of the 2 in complex 4. So for every 1 NADH molecule, we have 10 hydrogen protons pumped across the membrane. When it comes to FADH2, it isn't oxidized. It doesn't come into the electron transport chain until complex 2. So it bypasses complex 1. So it's only involved in the pumping of the 4 hydrogen protons at complex 3 and the 2 hydrogen protons at complex 4. So FADH2 helps contribute to the pumping of 6 hydrogen protons. NADH does 10. FADH2 molecule does only 6, and this is important to remember as we move forward when we talk about ATP synthesis. What I want you to see from this slide is for each mitochondria in a cell, we have multiple electron transport chains occurring in the inner mitochondrial membrane. Remember, the coenzymes that are produced from the oxidation of FADH2 and NADH2 by the electron transport chain are used by the citric acid cycle, which is in the matrix. So the proximity of these 2 pathways are very important. The next mini lecture, part D, will explore the production of ATP via the ATP synthase and the hydrogen proton pump.